Hello, welcome back to the Computational Genomics Lab. Today we are going to find peaks that are different between dox-treated and non-dox-treated samples. In the previous class we made a function to import our peaks, find the common peaks between all of them, and then overlap those peaks with gene annotations in the mouse genome. And so today we're going to go to lessons 09 and document 04 differential peaks. And so what we could imagine is if we took our 0 time points and did an overlap to say these are the peaks that are common in 0 and compared that to all the dox peaks, we could see which peaks were present in the 0 but not present in the dox-treated samples, or vice versa, peaks that were present that were no longer present in the dox-treated samples. So we're going to go to 04 differential peaks, and I'm going to load that here. And I will even clean my environment because we did that handy save in the last class. If you haven't done that, this would save you a lot of time. Here is I'm just going to source my functions, and I'm going to load all the results from the last thing where we overlapped all the common genes, and we have all these objects now of link RNA common overlaps, etc. So cool. We have all that. We have our my peaks, which is the most important one, which is our list of G ranges of each peak file. So we want to compare the 0 time points to the rest of these time points now. Cool. So to do that, we'll first make an object called non-dox samples by going into my peaks. And we're going to grab just the 2 0 time point samples that start with either this or wild type control or KO control, but have the 0 time point. So now I've subsetted that down to just 2 samples, and I can use the find common peaks function for that list of 2 G ranges here. And then we can see the length. There's about 53,000 peaks that are common in both 0 time points. Okay, so now to get all the other samples, which we're going to call dox samples, we can say pop open the names of my peaks, which is going to be these my peaks names that in our import peaks function were labeled by the file name. We're going to take those names and take anything that doesn't have 0, so all the other time points, 30, 60, 90, etc., and we're going to look for those in the names of my peaks. So when we do that, we will get a list of G ranges called dox samples, which are the files that start with KO, and there's a bunch of them, so we can just take a look at dox samples. Here we can see we have all the time points other than 0. Okay. So now what we'll do is take my peaks and only grab the dox samples. It's just another way of doing the same thing we got to, is we can say our dox peaks files are in our my peaks has all of our files. We're going to grab the ones with the sample name dox samples here, so not 0 time points, and to find common peaks with the dox peaks here. So what we're gonna we have the dox peaks here, so everything after 0, and then we can find the common peaks in those 2 samples now, and do that with our find common peaks function, and we can see there's 38,000 overlaps in all the other time points. So 2 time points were overlapped at 0, and 10 overlaps for the other samples that were just simply not 0. And that's looking at one of the samples. But okay, so let's do another comparison where we compare non-dox peaks, so that's the 2 0 time points, and dox common peaks, the peaks that are in all the dox samples, and do an overlap between those 2 with our find common peaks here. This should be interesting. So now we're saying what's in common between the 0 time point and the other time points. So if we look at the length of the docs common peaks versus those that are in both, it's roughly the same. So that's saying that if they're in the non-docs, they almost all overlap with the docs treated time points. So 37,000 out of 38,000 are in there. That's a large percentage. Spelled out a little more of that result here. In case that's confusing, you can pause it here, and it's teaching you the number of peaks that are changing between the 0 and the rest of the time points. So now if we look at non-docs common peaks, so those are the peaks that are in the 0 time point. If we look there, we see it's about 50,000. And in the overlap of those 57, it's like 20, like roughly, if we subtract these two, 16,000 peaks disappeared. So there was 16,000 peaks in the 0 time point that did not overlap with the non-docs control. If we did it the other way, you would not see anything. So what that basically means is all the peaks disappeared and no peaks reappeared, which is strange. And we're going to test this later. This is just more of an exercise of finding how you could do this with finding overlaps. And then in the next class, we'll use DESeq to use count tables for that. Okay, so here it's all spelled out what happened here, and the hypothesis this leads to is that there's 16,000 peaks that disappear. That are in the 0 time point that disappear when you treat with docs. So that could be interesting. Sounds suspicious though. Okay, so we are going to do make another function. I guess this is going to do pretty much the same thing our find overlaps does, but let's make a function called find my peaks and walk through that step by step. Okay, so we're going to take our common peaks, which are the peaks that are in all the samples. So you can say, here's the peaks that are in everything, and then compare that with the original peak files to see what was different. Okay, so we would do find overlaps between the original peaks and the common peaks. So now we're going to find all the things that don't overlap with the common peaks. We're going to get that out, and then we can find unique peaks by doing, this is a fairly complicated way of doing it, but we can take the original peaks and subtract out the overlaps that occurred in this find overlaps here. So we're going to say, take the original peak file and subtract out any of the ones that are in the common peaks and give us the ones that are just unique to that one file, and then return those unique peaks. Okay, so let's make that function, and then you can source it in your .r script. Okay, so then we're going to find my peaks, and so that's the new function we just made. That's going to tell us the unique ones, and we're going to compare the docs non-docs overlaps. That means peaks that are kind of common in everything. We got the same thing, several different ways of doing overlaps between all the samples, but in this case we separated non-docs 0 and docs, and then found the overlaps between them. That's this object here, and we're going to compare it with the non-docs common peaks. So we're going to find all the peaks that were in the 0 time point that are no longer present in the docs treated samples, and see if they all are really disappearing or what's going on there. Okay, so let's do find my peaks on those, and we got the 16,000 again. Okay, so these are 16,000 peaks that are only in the 0 time point and disappear. Okay, so now let's find the peaks that are unique to docs. And if we look at the length of that, it's about 1,793. Okay, so that means that there's this find my peaks is kind of misleading because it's actually finding the unique peaks between the comparison instead of the common peaks. So those are both super handy, not only to find things that are in common, but then to go back and find what was different in each sample. And that's what find my peaks is doing here. If we look here, we can now make we can look at how many of those overlap a gene that changed in our RNA-seq. So if we load, so now what we could do is say, okay, that's great that there's this many peaks changing, and you can export this as a bed file and look at it as IGV. This is more of an educational practice on overlaps, because when you do it with DE-seq, we'll see something more concrete. And we'll do that in the next class. So this is just basically saying where you can say what is unique in setting 1, what's common between 2 settings, what's unique in setting 1, and what's unique in setting 2, and have those tracked with G ranges. And the rest of this is here just to compare now the genes, the dox genes that disappeared, were they in our RNA-seq hits at all? So we did RNA-seq to say these genes changed, and we can now use our gene promoter thing to ask if the ATAC peaks are overlapping that disappeared onto those genes. Okay, so let's load our RNA-seq, and we're going to make a list of G ranges to run our find common peaks between gene promoters and non-dox promoters. So those would be ATAC peaks essentially that are unique to the non-dox, not the best naming here. But these are the things, the 16,000 that are unique to the 0 time point. And we're going to make an overlap between those. And now we're going to do the same thing where we do the find. Now we're going to find the common peaks here. So we set up our G range lists to do the comparison. And we see that 21,000 peaks overlap out of those 16,000 promoters. Okay, so now we can say, are those actually genes that change? Because that's a lot. So we're basically saying there's like a lot of peaks overlapping with gene promoters. So maybe those are the ones disappearing and going down in our gene expression. So we can take our SIG RNA-seq and take our non-dox gene overlap, so the 0 time point ATAC peaks that overlap genes, and we can say take those out that are in the filtered ResDF of significant RNA-seq genes using that in function. So it's just indexing 2 tables to intersect them and grab out just genes that were significant. And we can see that 47 genes do change that. We don't know if they go up or down yet. We could look at that next. And we'd want to look at those peaks in IGV. And so to do that, we would export these into our results folder as bed files, and those can be put into IGV. And you'll see a little box where there was an overlap with a peak at the 0 time point existed on 47 genes, and then that peak disappeared. So did those 47 genes go down? Might be interesting. But really we're talking very few, 47 out of 67,000 peaks. So not that many, which might be leaning to our answer that maybe dox doesn't affect the epigenetic state in just the gene expression state. Okay, so now we're going to do the next thing. We're going to do this more formally using DE-seq to ask what peaks change between the 0 time point and the rest of the dox treatments. Okay, we'll see you there.